Hello, everyone, and welcome to today's webinar, Current Trends in Cancer Immunotherapy, with a focus on cytokine therapies. I am Morgan Sterling of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Sino Biological. To learn more, visit sinobiological.com. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits after the webinar. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you're having trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Ritvika Biswas, Field Product Scientist, Sino Biological. Ritvika, you may now begin your presentation.
Hello, I am Ritvika and I'm a field product scientist at Sino Biological. Today I'll be giving a webinar on current trends in cancer immunotherapy with a focus on cytokine therapies. The entire webinar has been divided into 5 categories, and I'll start with a background of innate and adaptive immunity, followed by a brief introduction of immunotherapy, and then we are going to dive into cytokine therapy, and then we are going to talk a little bit about the cytokines for cancer diagnosis, and then we'll wrap it up with the challenges and future directions. So the first section is innate and adaptive immunity. The immune system is a complex system which is responsible for the protection of the human body. It consists of many cell types, structures, and chemical mediators with different functions that can regulate each other to work effectively and neutralize components recognized as non-self. The immune responses can be divided into 2 categories: innate immunity and adaptive immunity. The innate immune response functions as the first line of defense against infection. It consists of soluble factors such as complement proteins and diverse cellular components including granulocyte, mast cells, macrophages, dendritic cells, and natural killer cells. The adaptive immune response is slower to develop, but manifests as increased antigenic specificity and memory. It consists of antibodies, B lymphocytes, and CD4 and CD8 plus T lymphocytes. Delta gamma T cells and natural killer T cells are cytotoxic lymphocytes that straddle the interface of innate and adaptive immunity. Now let's discuss how the innate and adaptive immune responses work in cancer. Innate immune cells recognize cancer through germline encoded pattern recognition receptors and other cell surface molecules. Cancers express various stress-induced genes such as MICA and MICB that trigger receptors on NK cells, macrophages, and some cytotoxic T lymphocytes. NK cells also use a combination of inhibitory and activating receptors such as the KIRs to detect the loss of MHC class I expression on cancers. Dendritic cells phagocytose apoptotic tumors through CD36, and macrophages and DCs also use scavenger receptors and CD91 to ingest heat shock proteins complexed with tumor-derived peptides that are released from necrotic cancer cells. Now, adaptive immune cells are initially stimulated to recognize cancer cells through cross-priming by DCs. As tumor cells do not express co-stimulatory molecules that are important for T cell activation, they generally cannot prime cellular responses efficiently. Instead, DCs capture dying tumor cells or debris, migrate to the regional lymph nodes, and process the tumor-derived material into CD1D for presentation to NKT cells, and MHC class I and class II molecules for presentation to CD4 plus and CD8 plus T cells. Mature DCs express co-stimulatory molecules such as B7-1 and B7-2 to enhance T cell activation. DCs secrete IL-12 and IL-18 to promote T helper 1, CD4 plus T cell responses, and cytotoxic CD8 plus T cells. Activated CD4 T cells and NKT cells express CD40 ligand, which further stimulates DC maturation through CD40 signaling. CD4 plus T cells and DCs also help trigger B cells to produce antibodies that are reactive with tumor proteins. Now, following stimulation, natural killer cells can lyse tumors through perforin granzyme pathway or apoptosis-inducing ligands such as the TNF-related apoptosis-inducing ligand or TRAIL. NK cells secrete interferon gamma, which inhibits tumor cell proliferation, enhances tumor cell apoptosis, improves tumor antigen presentation, and inhibits angiogenesis. Now, NKT cells also execute cytotoxicity and cytokine production. Cytotoxic CD8 plus T cells lyse tumors through death ligands such as TRAIL and perforin granzyme pathway. CD4 plus T cells can differentiate into helper T cells, Th1 cells that secrete interferon gamma and TNF beta, or T helper cells 2 that secrete IL-4, 5, 6, 10, and 13. These cytokines enhance eosinophil function and increase antibody production by B cells. Now, antibodies to cancer cell surface molecules can inhibit oncogenic signaling and/or stimulate tumor destruction through engaging Fc receptors on macrophages, granulocytes, and NK cells. Antibodies can further promote tumor antigen presentation by dendritic cells through immune complex formation. Macrophages can lyse tumors through the production of nitric oxide and reactive oxygen species. So the gist of this section is that the body's immune system works to detect something harmful, producing antibodies and various proteins that fight the infection, and in this case, cancer. Now, with the general knowledge of innate and adaptive responses in cancer, let's move on to understand the basics of immunotherapy. So, cancer treatments have been improving for decades. Researchers are not only developing treatments that are more effective, but they are also highly focused on reducing their side effects. This is important given that in the United States, there are nearly 16 million people living with a history of cancer. That number is expected to increase to more than 20 million by the year 2026. Any problem that affects your healthy tissue and organs and arises as a result of cancer treatment is called a cancer treatment side effect. And side effects can vary from person to person and from treatment to treatment, to the extent that two people receiving the same treatment may experience very different side effects. Immunotherapy is a biological cancer treatment that boosts the body's own immune system to fight against cancer, leading to very few to no side effects. Immunotherapy works in various ways by stopping or slowing cancer cell growth and preventing cancer cells from spreading to other areas in the body. It helps a patient's immune system work better at destroying existing cancer cells developed in the body. So there are different types of immunotherapy. Essentially, immunotherapy types include checkpoint inhibitors, cancer vaccines, monoclonal antibodies, adoptive cell therapy, that includes T-cell transfer therapy, and immune system modulators. So let's quickly discuss these various types of immunotherapy. So one of the very common types of immunotherapy utilizes checkpoint inhibitors. This type exploits checkpoint proteins and other proteins that manage the flow of signals to T cells, telling the cells when to turn off and when to turn on. With this technique, T cells turn on to kill the cancer cells and they turn off so that they don't damage normal cells. Another type is cancer vaccine. These vaccines protect against cancer by helping the immune system identify antigens that are present on the cancer cells. Just like other kinds of vaccines, cancer vaccines use all or part of the cancer cells to help the body identify a harmful tumor in the body. Then there are monoclonal antibodies. These lab-made antibodies attack parts of the cancer cells and can also target cancer cells for special delivery of drugs, toxins, or radioactive material that can kill the cancer cells. Another very important type of immunotherapy is the adoptive cell therapy, also called T-cell transfer therapy. This treatment improves the immune system's ability to destroy cancer cells. Healthcare providers take a person's own immune cells and grow them in a laboratory. Once the cells have grown, results are injected back into the body so that they can kill the cancer cells. The last type of immunotherapy that we are going to discuss in detail today is the immune system modulators. Immunomodulators are substances that boost the body's response to cancer. Immune system modulators include cytokines, BCG, and immunomodulatory drugs. Now, immune system modulators are a type of immunotherapy that enhance the body's immune response against cancer. Most immune modulating agents are used to treat advanced cancer. Some are even used to help manage other side effects. The idea behind immunomodulation is that nominal poor immunogenicity of tumor cells results in part from their weak expression of MHC antigens, adhesion molecules, and co-stimulatory signals that could allow complete T-cell activation. Tumors may also secrete immunosuppressive molecules, and they often fail to express cytokines that activate local immune responses. These evasive strategies can be overcome by introducing molecules or genes for immune modulation into the cancer immunotherapy. Now, cytokine therapy is a type of therapy that uses cytokines to help modify the immune system to fight cancer. In cancer, cytokine therapy may help the immune system kill cancer cells and even keep them from growing. The two main types of cytokines used to treat cancer are interleukins and interferons. Now, let's discuss the role of cytokines in cancer and how we use that against cancer. So, the mixture of cytokines that is produced in the tumor microenvironment, or TME, has an important role in cancer pathogenesis. Cytokines that are released in response to infection, inflammation, and immunity can function to inhibit tumor development and progression. Alternatively, cancer cells can respond to host-derived cytokines that promote growth, attenuate apoptosis, and facilitate invasion and metastasis. Now, this diagram summarizes some of the major cell types found in solid tumor microenvironments. The action of the cytokines that have been exploited in clinical trials in patients with cancers on the various cell types are shown in red and green rectangles. Red rectangles indicate tumor-promoting actions, and green rectangles indicate anti-tumor actions. Some cytokines, such as transforming growth factor beta, TGF-beta, can have either anti-tumor or pro-tumor activity depending on the context. So in summary, the complex networks of cytokines and their regulatory pathways in all TMEs studied to date are shared across many solid tumor types, although the composition of these networks can vary depending on the immune landscape of the individual tumor type. Given that these cytokines play a role in activating, maintaining, and eventually terminating effective immune responses, but also their dysregulation in malignant disease, we will discuss both cytokine and anti-cytokine approaches to cancer therapy. Now, strong evidence indicates that some cytokines, especially those that are involved in adaptive immune responses, such as interferon gamma, IL-2, and IL-12, contribute to anti-tumor immunity. Over the past 40 years, attempts have been made to use these cytokines therapeutically. So now let's start with one of the most important cytokines, tumor necrosis factor. TNF is a pro-inflammatory cytokine produced by monocytes and macrophages, as well as by many other cells, including fibroblasts and endothelial cells, in response to stress. So this is an overview of TNF-alpha relevance in the breast cancer progression. Here we see in the diagram on the left panel that TNF-alpha is involved in all stages of breast cancer progression, being involved in cell proliferation, survival, and motility, inflammation maintenance, acquisition of stemness, and resistance to chemotherapy. On the right panel, we see that TNF-alpha is secreted in the TME by various cell types, including macrophages, lymphocytes, fibroblasts, and tumor cells, and impacts the fate of the primary tumor by acting on both tumor and the stromal cells. In a phase 1b clinical trial, 14 patients with advanced stage melanoma were treated with nivolumab and ipilimumab, plus either infliximab or certolizumab. Both combinations were found to be safe and to have clinical and biological activities, including systemic T-cell activation. At Sino Biological, we have a highly cited and optimized recombinant human TNF protein available that has been validated for activity, verified by HPLC, and now we also have GMP-grade recombinant human TNF alpha protein product available. Now, let's talk about interleukin 6. IL-6 is a pleiotropic cytokine involved in the regulations of various physiological and pathological processes. The IL-6 JAK-STAT3 signaling pathway has major immunoregulatory roles in health and is dysregulated in many diseases, including cancer. IL-6 causes uncontrolled inflammatory responses, resulting in chronic inflammation and even carcinoma, and its expression is associated with poor prognosis for breast cancer. IL-6 antagonists such as tocilizumab are commonly used to alleviate severe CRS that occurs in some patients treated with CAR T cells. 3 IL-6 antagonists are clinically approved and commonly used. Ciltuximab, which is an anti-IL-6 monoclonal antibody, tocilizumab and serulumab, which are anti-IL-6 receptor monoclonal antibodies. At Sino Biological, we offer an optimized, untagged, and highly cited recombinant human IL-6 protein. We also offer this protein for various species and with different tags. Now let's talk about interferon gamma. Interferon gamma is a major regulator of the innate and adaptive immune system. Originally identified based on its antiviral activity, this cytokine is produced by NK cells, NKT cells, CD8 plus, and CD4 plus T cells during innate and adaptive immune responses, often in response to IL-2 stimulation that targets macrophages and B cells in response. So I'm showing you the mechanisms of interferon gamma mediated cancer regression on the left panel here. The interferon gamma stimulates activation of JAK-STAT signaling pathway in the TME that further drives increased caspase activity. Consequently, cancer cells undergo apoptosis. Interferon gamma induces fragility of regulatory T cells and inhibits the formation of tumor blood vessels by downregulating VEGF-A. Through stimulation of M1 macrophage polarization and inhibition of their M2 phenotype, it contributes to effective anti-tumor immune response. Moreover, interferon gamma increases FN1 expression that limits cancer metastasis. In patients with ovarian cancer, intraperitoneal administration of interferon gamma was associated with substantial tumor regressions. Sino Biological offers high-quality recombinant human interferon gamma protein, and this product has been cited over 24 times. Now let's talk about interleukin-2. IL-2 is a protein that plays an essential role in the development and function of a spectrum of T cells, including regulatory T cells. Consequently, IL-2 has numerous antitumor effects, including stimulating T cell and NK cell proliferation and function. The immunostimulatory properties of IL-2 provided the rationale for clinical trials initiated more than 30 years ago to evaluate the anti-cancer activity of this cytokine. Sino Biological offers an optimized recombinant human IL-2 tag-free protein with over 95% purity and high bioactivity. Now let's talk about another widely used cytokine, IL-10. As seen on this diagram on the left, systemic PEG IL-10 may bind either CD8 plus T cells or other IL-10 receptor leukocytes in the TME, following an increase in the intratumor CD8 plus T cell numbers by a number of possible mechanisms. IL-10 is generally considered to be an immunosuppressive cytokine in the TME owing to its broad anti-inflammatory effects. However, IL-10 has anti-cancer effects in various tumor models through increased CD8 plus T cell activation. A series of trials have been performed using pegylated IL-10 preparation called peglodecakin in patients with advanced stage solid tumors. This monotherapy was reported to increase the number of activated infiltrating CD8 plus T cells in tumors with low T cell infiltration prior to therapy. Sino Biological offers high-quality recombinant IL-10 proteins for various species as seen here, such as mouse and human. Now let's talk about interleukin-12. IL-12 stimulates NK cell and CD8 plus T cell proliferation and increases their cytotoxic activity. In addition, IL-12 promotes Th1 cell differentiation and interferon gamma production, thereby enhancing antigen presentation. So this diagram on the left panel illustrates in vitro IL-12 conditioning of CD8 plus T cells. So IL-12 binds to IL-12R beta 2 and then induces a series of events of cell activation mediated by tyrosine kinase 2, JAK2, and STAT4. These events result in an overall generation and activation of CTLs. Priming of naive CD8 plus T cells in the presence of IL-12 selectively rescues early activated CD62L expressing CD8 plus T cells from activation-induced cell death and results in increased accumulation of this subset of CD8 plus T cells and mediated anti-tumor effect upon their homing to the tumor sites. Systemic IL-12 has been shown to promote tumor infiltration by CD8 plus T cells, at least in patients with melanoma. To circumvent toxicities, NHS IL-12 was developed through the fusion of two IL-12 molecules to an antibody targeting DNA histone complex, which enables selective delivery of the cytokine to the necrotic tumor regions. More recently, a trial of intratumoral plasmid-derived IL-12 in combination with pembrolizumab in patients with immune cold melanomas, which are unlikely to respond to anti-PD-1 mAb monotherapy, showed great promise. Sino Biological offers high-quality recombinant human IL-12 tag-free protein with over 95% purity and high bioactivity. Now let's talk about interleukin 15. So myeloid cells present interleukin 15 in association with IL-15 receptor alpha to the heterodimeric complex of IL-15 receptor beta and the gamma chain on CD8 plus T cells and NK cells, which recruits and activates JAK1 and JAK3, which finally results in the activation of mTOR. This activation of mTOR triggers metabolic reprogramming to the glycolytic pathway and manifests various cellular responses in CD8 plus T cells and NK cells that promote killing of microbes and anti-cancer immunity. In preclinical models, IL-15 is another cytokine that promotes the generation, proliferation, and activity of anti-tumor NK cells and CD8 plus T cells, but not of tumor-supporting Treg cells. The effects of this cytokine in preclinical models of cancer and oncology clinical trials have been reviewed extensively. More than 170 trials of IL-15 have been initiated, but all of these have shown very limited efficacy and monotherapy despite substantial beneficial effects on circulating NK cells and CD8 plus T cells. IL-15 has a very short half-life, but is much more stable when bound to its high-affinity receptor IL-15RA. Therefore, several so-called IL-15 superagonists consisting of IL-15 in complex with truncated forms of IL-15RA have entered clinical development with promising initial results. The most extensively trialed superagonist is called N803, has been tested in combination with other immunotherapies. At Sino Biological, we offer recombinant IL-15 protein as well as superagonist IL-15 and IL-15RA. The recombinant IL-15 protein has been validated for high bioactivity and has a purity of over 95%. The superagonist combination of IL-15 and IL-15RA has been validated by SEC-HPLC with a purity of over 90%. Now let's talk about the last one in the list, that is interleukin-1 beta. So the figure on the left panel illustrates the functions of IL-1 beta signaling in cancer. The IL-1 signaling pathway is activated through the binding of IL-1 alpha or IL-1 beta to the IL-1 RI. Agonist binding is followed by engagement of further signaling intermediates with subsequent activation of transcription factors such as NF-kappa B and AP1. Transcription of inflammatory genes is upregulated, which may then result in pro-tumorigenic or anti-tumorigenic phenotypes. Now, similar to TNF and IL-6, a large body of preclinical data underscores the tumor-promoting effects of IL-1 alpha and IL-1 beta. IL-1 beta is mainly produced by myeloid cells and fibroblasts in the TME, especially via the inflammasome pathway, and can stimulate other inflammatory mediators, cell invasiveness, and immunosuppression. Clinical trials evaluating the anti-IL-1 beta antibody, canakinumab, with various combinations of cytotoxic agents and/or immune checkpoint inhibitors are underway. Preclinical evidence indicates that inhibiting IL-1 might alleviate the neurotoxicity of CAR T cell therapy, and a first clinical trial of anakinra for this indication demonstrated promising activity. At Sino Biological, we have developed a highly optimized recombinant human IL-1 beta protein that is available and has been validated for activity with purity over 95%. Sino Biological offers both research-grade and GMP-grade cytokines. GMP-grade cytokines are manufactured in compliance with GMP standards, which involve a strict quality management system, standardized quality assurance management, and quality control system. These systems are designed to minimize variability and uncertainty in the manufacturing process, ensuring high quality of cytokine products. Sino Biological is dedicated to producing high-quality reagents for cell therapy research and clinical applications. Utilizing advanced GMP-grade quality management system and releasing testing criteria, Sino Biological has successfully developed a series of high-quality GMP-grade cytokines. The advantages of using Sino Biological cytokine products include high quality, HPLC verification, high purity, high bioactivity, low endotoxin levels, and tag-free cytokines that are produced in-house and offered at very competitive prices. Here on the left is a list of the GMP-grade cytokines that we carry at Sino Biological. Now let's move on to our next section of cytokines for cancer diagnosis. Multiple cancer-derived proteins have been established and used at clinics as biomarkers to detect cancer from blood. However, other approaches include detection of a set of cytokines, combining cytokines and their soluble receptors, and combining cytokines and cancer proteins may provide better accuracy for cancer diagnosis and prognosis. A highly accurate alternative cancer biomarkers have been proposed, which is the cytokine secretion profile of the circulating peripheral immune cells. Here is an overview of the reagents for cytokines in cancer diagnosis offered by Sino Biological. These are some of the selected examples for cytokine biomarkers for cancer diagnosis. As you can see, these antibodies have been verified for various applications such as ELISA, Western blot, immunohistochemistry, immunofluorescence, neutralization, flow cytometry, etc. Sino Biological carries over 600 ELISA kits, over 14,000 antibodies and antibody pairs against multiple species. And if you cannot find the antibody of choice in our catalog, we also offer customized antibody production services. Now, as always with all the different therapeutic approaches, cytokine therapy also has some challenges. In this next section, let's discuss some of the challenges along with future directions. So on this slide, I'm listing some of the challenges of cytokine therapies. The biggest challenge of cytokine therapy is the pleiotropic nature of different cytokines. The left panel lists out the different challenges posed by cytokine monotherapies, such as the very short half-life of cytokines in circulation in the body, poor biodistribution of the cytokines, and pathway redundancy of the target molecule. There is also a very narrow therapeutic window with prohibitive cytotoxicities and the other immunosuppressive effects of cytokines, along with context-dependent effects of cytokines. On the right panel is a list of challenges posed by cytokines or chemokine agonist therapies. Some of the biggest challenges are malignant cell evolution over time, which results in variations in the immune landscapes, immunosuppressive actions of agonists in general, and the cytokine pathway redundancy, which may lead to side effects. Undoubtedly, cytokines have proven to be effective in cancer therapy. However, the effectiveness of some promising targets on various immune cell populations remain poorly understood. The current direction of clinical research with cytokine-based therapies is focused on either their combination with other immunotherapies and/or modifications of cytokines to alter their pharmacokinetics and binding affinities in an effort to reduce toxicity and avoid augmentation of immunosuppressive factors such as Treg cells. New approaches that improve targeting of cytokines and alter their pharmacokinetics may be useful, such as cell-based or other vector delivery, chemically modified recombinant proteins, etc., to overcome limitations of different cytokine therapies. Current trends in the development of cancer immunotherapy indicate that cytokines may find their greatest role in therapy when administered in combination with other agents, such as immune checkpoint inhibitors, oncolytic viruses, or as a component of DC-based and tumor cell-based vaccines. Some researchers are also incorporating cytokines into oncolytic viruses or CAR T cells, allowing them to produce useful cytokines within the tumor sites. So, I will wrap up this webinar with an overview of Sino Biological. Sino Biological is an internationally recognized reagent supplier and contract research organization with focuses on recombinant protein and antibody services. As of now, we carry a large number of catalog products, including over 6,500 proteins, 14,000 antibodies, over 47,000 genes, and over 600 ELISA kits. Up to September 2022, our products have been cited in over 10,000 publications, and some of these include high-impact journals such as Nature, Cell, and Science. Also, we are a ISO 9001 and ISO 13485 certified company, supplying reagents to researchers in over 90 countries worldwide. Sino Biological has multiple offices throughout the globe, with headquarters being in Beijing, China, along with other office spaces. We also have other offices in Japan, Germany, and 2 offices in the United States, one being in Pennsylvania and the other one in Texas. Very soon, we are opening an R&D facility in Houston, Texas, called the Center for Bioprocessing, that will serve as our CRO production facility here in the United States. Thank you very much, everyone, for accompanying me on this webinar.
Thank you, Ridwika Biswas, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located at the far left of your screen. We'll answer as many questions as we have time for. So let's get started. Our first question is, in terms of cancer biology, what are the most common pathways involved?
Okay, so thank you for the question.
So some of the most important pathways involved in cancer biology are the ER beta family pathway, the p53-mediated apoptosis pathway, and the GSK3 signaling pathway.
Awesome. Thank you. And a follow-up question to that, in terms of cytokine therapy, what are the most common pathways that are targeted?
Okay, that's actually a great question. So the effects of individual cytokines on immunity depend on several factors, including the local cytokine concentration, the expression pattern of the cytokine receptors, and the integration of multiple signaling pathways in the responding immune cells. Now, if you ask for specific pathways, the most important pathway involved will be the JAK-STAT pathway, because most of the cytokine receptor signals through the JAK-STAT pathway. So basically, a good understanding of the molecular signaling pathways used by cytokine receptors and the temporal pattern of the receptor expression are critical in the development of effective cytokine-based cancer treatments.
Great. And why do cytokines have pleiotropic effects? How is pleiotropism important in immunotherapy?
Okay, thank you so much for the question. So cytokine signaling is characterized by a significant degree of pleiotropism, in which one cytokine has the ability to act on many different cell types to mediate diverse and sometimes opposing effects. And that's what we saw basically on the slide where I talked about the actions of cytokines in tumor microenvironment, where some cytokines have both anti-tumor and pro-tumor effects. And this has proven to be one of the primary limitations to IL-2 therapy due to the dual function of IL-2 as a potent activator of the T effector compartment as well as the T regulatory compartment. Another important property of cytokine signaling that I did not really talk about is its degree of redundancy, in which multiple cytokines have the same functional effects. And this redundancy can make the therapeutic manipulation of cytokines somewhat challenging, since modification of one cytokine can be compensated by others. And this extensive pleiotropy and redundancy of the cytokine signaling mainly suggests that the future of cytokine-based therapy may be in combination regimens that target multiple pathways to amplify the anti-tumor response while suppressing the regulatory pathways and minimizing toxicities. And for an example, like in the targeted therapy using BRAF inhibition, an application of cytokines in combination with these therapies is actually generating quite a lot of enthusiasm recently. And cytokines have already proven to be effective in the treatment of cancer. And there is very little doubt that they will continue, to play a major role in the development of cancer immunotherapy.
Great. So we saw multiple GMP grade products by Sino Biological. Do they have citations?
Oh, yeah. Actually, almost all the GMP grade cytokine products have been cited in various journals. So let me quickly discuss this one citation for our GMP grade IL-2. This paper was published in Frontiers in Oncology, and this was titled Characterization of the Therapeutic Effects of Novel CAR-T Targeting CD38 on Multiple Myeloma. And here in this paper, they used our product to activate the peripheral blood mononuclear cells, in short PBMCs, that were isolated from healthy human donors. And the findings of this paper were that a novel CD38 targeted second generation CAR T cells have efficient and specific anti-tumor activity, and it may actually become a novel therapy for clinical treatment of multiple myeloma. So yeah, our GMP grade cytokines have been cited in papers with important results and implications so far.
Amazing. So you talked about cytokines being used for cancer diagnosis and prognosis. Can you elaborate on that with an example?
Yeah, sure. So the dysregulated circulating cytokines are not the only immune biomarkers that can be detected in the circulation during cancer. Instead, in cancer patients, peripheral immune cells have dysregulated immune cytokine signaling signatures. And there have been some recent studies that show dysregulated IL-6 secretion from peripheral T cells and dysregulated interferon gamma secretion from peripheral monocytes, even from patients that had localized tumors. And in breast cancer patients, the dysregulated cytokine signaling signatures in the immune cells strongly predict the risk of future relapse. And I think it was done in like 2 independent breast cancer cohorts, which point to cytokine responsiveness as biomarkers to evaluate the immune status in these patients.
Awesome. Great. Thank you. Looks like we have time for one more question. So, are Sino Biological cancer diagnosis antibodies carrier free?
Yeah, so if you're looking for the short answer, then yes, cytobiological cancer diagnosis antibodies are carrier free. And if you're looking for the long answer, we have over 14,000 antibodies. And to enhance flexibility, we have formulated 100% of our antibodies and matched antibody pairs to be carrier free and conjugation ready. The antibody can be directly added to the conjugation reaction, avoiding antibody loss without the need to perform any buffer exchange.
Thank you so much, Ridwika. Do you have any final comments for our audience?
Yeah, actually, so if any of the topics covered here interest you, please check out the QR codes here. And if you have any questions, please don't hesitate to reach out to us at sinobiological.com. Thank you.
Thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Sino Biological, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We do encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you so much. Goodbye.